complex systems like societies and ecosystems are never stable. They just always change, and usually that change has the character of slow trends and fluctuations around them. But every once in a while, a big thing happens. There is a sudden shift in the state. And here is an example from an ecosystem where we have been studying this little creature, the brittle star at the bottom of the ocean. We follow it for years, and suddenly you see it slips into a very different state, this ecosystem, and this organism becomes rare, and it has been rare ever since. Well, this kind of transitions you see in all kinds of ecosystems every once in a while. And also, if you look further back in time, you see that life on Earth has evolved, has changed, but also those transitions have occasionally been really radical, as in the case, of course, as the well-known extinction of the dinosaurs that made space for mammals such as us. So if you see that kind of radical change, you always wonder, why did it happen, right? What caused it? And the natural thought is at first that there was a big perturbation that caused the system to really change radically. And that may often be the case, as in the case of the dinosaurs. There was a big perturbation. But there is also another possibility when you want to explain radical transitions, and that is the idea that there was a tipping point. The idea that the system became so fragile that in the end you didn't need a big perturbation, but just a tiny little push would already initiate a radical shift to a different state. And a well-known way to depict that is with those ball in cup, marble in a cup diagrams that surely you have seen on another occasion. So as this is the basis of the theory that I want to talk about, I'm going to take you through this little graph just briefly. So what you see here is a graph of the system state as a function of conditions. So for instance, this may be the nutrient in stream in a lake, and this may be how clear the lake is. And systems that have this particular behavior, this folded graph, they behave in a strange way. Because what you see is that if you start here and you change the conditions, the state actually hardly changes until you reach a point where it radically changes. And you can understand that from those stability landscape. Here, there is only one state. If you perturb the system, it'll roll back. If you change the conditions, the system will stay basically in the same state. It looks like nothing really changed, but something did change because the landscape of stability around the system changed. And here, this valley of attraction has become small so that even a tiny perturbation at some point may invoke a shift to the alternative state. And once you have shifted there, even if you reverse the conditions, you stay in that new state. So this graph tells you the equilibria of the system. The solid line is a stable state, and this equilibrium, the stippled one, is an unstable one. That's the hilltop here. And you see that the hilltops and the valleys are part of this bigger figure. And what happens if you change the conditions is that this valley, the stable state, may be remaining the same, but something is coming closer, and that's the hilltop. And in the end, they fall together, and the whole thing loses stability. So we published the paper with this graph in 2001. in nature, and it has been cited thousands of times since then. But it wasn't new. It was an old idea. And you can see that in this picture, the last photograph taken of the famous painter Salvador Dali. And if you look closely, you see that he is posing in front of a painting, his last painting in life, which is exactly the same figure. And that's not a coincidence. Salvador Dali was very interested in the theory of catastrophe, the catastrophe theory, as it was called by mathematicians in those days. He had heard of it, and he talked to famous mathematicians, even organized meetings to get them together. And at the end of his life, when he wanted to make one more dramatic statement, he dressed up one more time, had his last picture taken in front of his last painting, pointing to, I think, what he thought was one of the most profound things he had learned in life, and one of the most relevant things at this point in time, because he felt he was actually reaching his own tipping point. So let's talk about why that old theory suddenly became popular again, and now you see lots of literature, lots of new research on it. The main reason is that in the 60s, it was basically a mathematical theory that could not really be connected to the real world. Whereas now we're finding applications in all kinds of sciences, ranging from climate science, where you hear a lot about climate tipping points, to ecology, to psychology. And I'll explain a little bit about that in this lecture. So one way of connecting this theory to the reality is using big data, using data to, for instance, construct this kind of stability landscapes. And to see how you can do that, you have to imagine that this ball is not just laying in the deepest point, but it's all the time pushed by invisible hands, so to say. It's real life. There's always good years, bad moments, perturbations that make the ball flipper around. And this combination of the stochasticity of the world that affects the state, while at the background you always also have this overall stability properties, make that the change is of course not random in the movement of the ball. You clearly see that it tends to stay around this state of the valley longer than, for instance, in the middle, because this is unstable, it's a transient state. And here it also stays, but not as long as in the more resilient state. So this information, this behavior of the system, you can, for instance, simply make a probability density, just a histogram of the states. And if you make a histogram of the states, the averaging over all this information, you get a lot of states here, less here, and a lot there. And you see, if you flip that, you basically get this stability landscape. So I explain it in a very simple way now, but there is a more formal mathematical theory about that. Now, this is beautiful if you have this fantastic long time series to probe the system. But very often we do not have such long time series. But instead we may have snapshots. For instance, if you're looking at, let's say, the microbiome of a person, maybe you don't have this very detailed long time series, but maybe you have snapshots of a microbiome of many persons, maybe hundreds or thousands of persons. I'll talk a little bit more about that example later, because we've actually done that. But I'll start with another example of using snapshots that we applied in our studies of ecosystems. So one of the big ecosystems on the globe is the tropical rainforest. And the tropical rainforest has this property that it can flip into an alternative stable state, which is savanna. You rarely see something in between, like you tend to see either lots of trees or you have this open landscape with a very limited number of trees dominated by grass and kept stable by fire. So the way in which we studied that was using remote sensing to look at many places, many little spots on Earth. What was the tree cover? And then we looked up what was the average rainfall in that region. And you see, if there is a lot of rain, you'll see a lot of trees. This is the rainforest. And when there is no rain, there is very little trees. And in between, you can have this other blob of intermediate tree cover, which is the savanna. So combining the information of mean annual precipitation and tree cover, you can use this point density to compute those landscapes, those stability landscapes, showing you that, for instance, the rainforest is separated by this unstable equilibrium from the savanna. This unstable 50% tree cover is unstable. You either go into forest, it's a self-reinforcing change, or you slip into savanna through a fire feedback. And there is this other state, the dry, the treeless state. Now, let's think about the microbiome. Rain is obviously not a very interesting variable to look at. Food maybe, but there is another one, and that is time. So if you look at any physiological system, and you look at the resilience of humans and their subsystems, time is important. Nobody escapes time. We're all aging, and as we age, we see that the stability of different systems changes. Now here is the example I mentioned that we did of the human gut microbiome. It's work by Leo Lachty and Willem de Vos and others. Those are the bacterial counts of a particular group in healthy humans. And what you can see is that young adults tend to have a low density of this group, elderly a high density, and in between it can be either of the two. So this kind of techniques can be used also to study the microbiome. What is I find fascinating when you study humans or animals is that time is actually a strange thing. The time doesn't run strictly the same for everyone, right? If you're lucky, it runs slower for you. This is a painting by Salvador Dali, by the way. And one way to depict it is that we all have this clock ticking. We're all aging, but it's not ticking the same rate for everyone because it depends on your lifestyle, right? If you exercise and eat healthy, it depends on your genes. You can be lucky with your genes. It depends on life events. And all those things together affect the complex system that we are. If you're interested in this view, it's published in PNAS, and the article is called Systemic Resilience of Humans and Other Animals, because it's a cooperation between medical scientists and people working actually on chicken, on cows, on nematodes. It's basically the same story in all those animals. So how do we work as a complex system? We have subsystems like the brain, the clear subsystem, the heart, the liver, the arteries. The microbiome is also a subsystem. And those subsystems work together to keep critical parameters within the right limits. The temperature, your glucose level, your posture, make sure you don't fall, your moisture balance, your mood. So this complex system is basically what we want to understand. We want to understand human resilience. We want to understand aging, and whatever your special field is, I think it's interesting to try to understand how it fits in the whole field. And I find it really exciting that the microbiome research very much is taking this road, that it looks at the brain-gut connection, for instance. So how do you measure this kind of things? Salvador Dali clearly had an intuition for it. He felt that when he was young, he was just different. He was more resilient. Now he felt different. He felt that when he was young, anything could be pushed against him, but he would be firmly in place. Now, even a small thing could push him out of the small valley that kept him alive. But how, if we don't have that intuition, how can we scientifically measure resilience? How can we measure if a system is becoming frail? Is there a way to do it? Is there a way to do it for complex systems in general, like humans, societies, the climate? The reason we started thinking about that as a group of theoreticians, of mathematicians basically, is that we knew that in any complex systems, when you reach a critical point, universal laws start to rule the dynamics. And that makes it possible to dream that perhaps the same fundamental principles, same fundamental signals can be used to see if a system is nearing a tipping point, which is of course really exciting, because we would like to know if not only the climate system, the thermohaline circulation, but also, for instance, the financial markets are reaching a tipping point, and so forth. So the line of thinking here is that we can derive early warning signals if we find indicators of resilience, generic indicators that show if a system is becoming unstable. This work is the result of a cross-disciplinary workshop, lots of walking in the dunes here in the Netherlands where I live, with economists, mathematicians, animal scientists, and so forth, scribbling on flipovers, the thing we miss so much now that we are meeting virtually all the time. And the end result was this paper, a paper published in 2009 in Nature that now has been cited thousands of times and created an avalanche of other papers that are about the same topic. These are just only papers in Nature and Science, and there are many, many, many more. So what was the fuzz about? What's the basic idea? It's very simple. The idea is that if you are in a resilient state and you're pushed, you recover quickly. This slope is steep. But if you are in an unresilient state and this value is disappearing, it's becoming very tiny, very shallow, then the slope is not steep, and small perturbations, the recovery from small perturbations becomes very slow, eventually becomes zero. And that phenomenon is known already for a long time in mathematics as critical slowing down. The system is becoming slow to recover because it's reaching this critical point. So this is the idea. If you are thinking again of this catastrophe fault, this S shape, if you are far from the critical point, you recover quickly. if you're close to the critical point, it takes longer. Now, this supposes that you can actually give a little push to see if it recovers quick. And often we can't do that. We can't do that with the climate system, for instance, to see if part of the climate system, such as a thermohaline circulation or an ice cap, is close to a tipping point. We can't do that. And also ethically, we can't do it, for instance, with people. We can't push their mood a bit to see if they fall into a depression. It's not ethical. So are there other ways to infer this information? Yes, there are, because there is always little pushes. The climate system is always pushed by like little volcano outbreaks, and so your mind, your mood is always pushed by things you experience. I'm in this lecture perturbing your mind a little bit, and as a result of that, all systems fluctuate all the time. Now, given the same regime of perturbations, theory predicts that if the system becomes low resilience, the fluctuations are different from when they are high resilience. And what you see in this simulated example is two things. One is that the variance increases, but more importantly, it becomes slower. And how can you measure that? You can measure that from the temporal autocorrelation. If you plot the state now against the state in the next moment, you'll see it becomes more correlated than in the resilient system. So this increase in autocorrelation can be a signal that the system is becoming unstable, that it's approaching a tipping point. Two indicators that we call DORs, dynamic indicators of resilience. Dynamic indicators because they really probe resilience. It's not like an indicator of health, like how pale you look or what is your temperature. No, you study the micro recoveries of the system on the perturbations that happen all the time. And this gives you the information like the recovery rate and the information that you have in autocorrelation and variance. And there is one more thing that I would like to mention, and that is that if you study a system like a human consisting of many subsystems, like the brain, the intestines, and so forth, it's also interesting to look at how those subsystems interact. Are their ups and downs correlated or not? You can imagine that for instance, if your mood is stable, nothing happens to it. But if it is becoming less stable, if you hurt your knee, maybe it affects your mood quite strongly. And maybe that affects your eating pattern. And maybe that affects your intestinal microbiome. So you'll see that if the different subsystems are becoming less resilient, they will be affected more if one of the subsystems is becoming in disarray. So that's all very nice. Those are all mathematical predictions from the theory, but is there any evidence that we can really find these signals? Well, there is. And the first system we looked at was the Earth system. Why? Because the Earth system, we know that the Earth system not only changed gradually, but every once in a while there was a big change. This is the example of the greenhouse Earth ending. The Earth didn't have ice caps for most of its life until only 34 million years ago. Here is the time scale when we got ice caps, and they remained ever since. Could you see that coming? If you were a scientist and you saw this, what would your prediction be? Would you expect this? Probably not. Unless, of course, you knew the theory of critical slowing down and you had measured autocorrelation. You would see something was brewing up in the system autocorrelation was increasing. Here is the other example of the end of the glaciation. What would your prediction be if you didn't know the future? Would you predict this? Well, maybe if you had heard this lecture and studied this indicator of critical slowing down. We also looked in the lab. We made ecosystems in the lab. It's the work of Annelies Verraert. This is our micro ecosystem of cyanobacteria that we drove to a point of collapse. And Annelies showed that also there you could see the predicted early warning signals. And then we found it in remote sensing time series of tropical rainforest. People found it in Neolithic societies before they collapsed. We found it in the mood of humans. This was a project where we tried to predict how likely people are to fall into a depressed state later in life. And we used time series of mood fluctuations. And this is the work of Ingrid van der Leemput. What she showed is that you can actually quite well predict how much risk people are to fall into this shift in another mood state later in life from those micro fluctuations in the mood. And also it's interesting, once people are depressed, you can also predict whether they're more likely to flip out of the depression also from those small fluctuations. So yes, he was right. Salvador Dali felt there was something really deep going on here. There was a deep universality, and he was right about that. He was also right about his intuition that this deep universality became especially relevant at this point in life for him. With respect to the conference that we are having, I think the most interesting thing would be to see if we can have a discussion about to which extent we might use this kind of thinking, those results, studying the microbiome. And so I've been thinking a little bit about that, and questions that came to my mind were, are there actually contrasting alternative stable states in the microbiome? I think there are. I think indications, for instance, are the behavior of the gut microbiome with the Clostridium difficile invasion. You see that the system can flip into this state dominated by this bacteria, and it's very difficult to get it out unless you reset the system by a transplant with a healthy microbiome, and then it tends to remain stable. That's now well established, and it is kind of a proof that there are two alternative stable states, and you can flip the system back to the original state by a perturbation. Also, the work that we did with Leo Lahti on bacterial groups, just looking at their abundance, shows that bimodality, particular groups tend to be either very abundant or very rare, and that is also suggestive of, even within the healthy population, alternative states like flip-flop possibilities being there. And if that is the case, I think it's really interesting if we can think about ways to predict critical transitions, for instance, out of the healthy state into a diseased state. Can we use this kind of indicators, quantitative indicators, based on fluctuations in the system or on measurements across many individuals? Can we use that to estimate resilience? And if that is successful, I think it would be really cool to see if we can use that then to study the effects of diet and age and interventions on the resilience of different microbiome states. Because if you can really measure resilience, it's a much more solid way of looking at effects of all kinds of factors. That's basically what we're also doing in our research with geriatricians on elderly people, trying to, for instance, measure the resilience of the balance system by putting people on a balance plate with sensors and measuring microfluctuations. We can then directly see effects of sway. You see really the critical slowing down there. And we can do training interventions, for instance, of people, muscle strengthening interventions, and see if you can affect the resilience. So you have an endpoint that you can measure then. And of course, this whole idea of the cross correlations, and if we think of the microbiome, if only as one of the subsystems that makes up a human being, can we get information out of fluctuations in the microbiome in relation to fluctuations in the brain, in other systems that we have? So those are, I think, there is a lot of open questions, and I would say high risk, high gain, perhaps possibilities. And I would like to close my lecture with this and invite you to the discussion section and see if we can have a nice conversation about this. Thank you very much.